asked by the that can be asked by the UPSC in this syllabus. Now, till now we have discussed about we have till now we have discussed. Hello, am I audible? Yeah, thank you. So, till now we have discussed about the basic concepts of biotechnology. Well, we have learned about the genes, we have learned about the chromosomes, and we have learned about the DNA and the other things, such as we have also talked about the stem cells, we have talked about the fertilization, we have talked about the twins, that the formation of twins. Now we have also learned about the ploidy. Today we'll start our discussion with the aberrations. Now aberrations are something that is not normal, is the aberration. So the aberrations are of two types. Like genetically, we have two types of aberrations. We can have two types of aberrations. That is the chromosomal aberrations. That is the change in number of chromosomes. Chromosomal aberrations means a change in number of chromosomes. That is the chromosomal aberrations. Then we also have the autosomal aberrations. That is the change in the number of autosomes. So this is chromosomal aberrations is the change in number of sex chromosomes, and the change in number of autosomes, that is the non-sex chromosomes, are the autosomal aberrations. Now, a normal genotype, if the genotype is there are two X chromosomes, the two sex chromosomes are the two X chromosomes, then we get a normal female. Now, the total number of chromosomes here will be 46. So the total count of chromosomes in a normal female is 46. Now, sometimes due to some unforeseen factors or circumstances, the number of the sex chromosomes, they increase in an individual. So instead of two X chromosomes, now this particular female is having three X chromosomes. So we call it the super female syndrome. In normal female, we have two X chromosomes, and in super female syndrome, we have a female with three X chromosomes. Now, this is super female, but this female is sterile, or we can call it infertile. And can you guess the total number of chromosomes that this particular female will be having? The total number of chromosomes here will be 47. So we have 44 autosomes plus we have two sex chromosomes. Okay. If out of these two sex chromosomes, both of them are X, then we have a female, that is a normal female. But instead of these two sex chromosomes, if we have three sex chromosomes and there are three X's, so we say that it is super female syndrome, and the female is sterile or infertile, and the total count of chromosomes increases to 47. Now, if the two sex chromosomes, one of the chromosome is X chromosome and the other chromosome is the Y chromosome, then we get a normal male. So if the genotype is one X chromosome and one Y chromosome, then we get a normal male. That is, the male is having 46 chromosomes. Okay, a healthy normal male having 46 chromosomes. But as in the case of females, if an extra copy of this Y chromosome, if the extra copy of Y chromosomes is added to the genome, then we say we have got a super male syndrome. So there are three sex chromosomes, two of them are Y. So that is the super male syndrome, and the total number of chromosomes here will be 44 plus 3, that is 47. 44 autosomes plus 3 sex chromosomes, that is 47 chromosomes. Now, that is like till now we have discussed about the addition of an extra chromosome. What if A chromosome, the second chromosome, that is the second sex chromosome, it can either be the X chromosome or it can either be the Y chromosome. What if it doesn't form? So the individual is left with only a single sex chromosome. So in such a condition in which we have a single sex chromosome, that is the X chromosome, the second chromosome, that it can be the X or it can be Y, but now it is dropped or it has not, the organism, the individual has not got this second chromosome, then we say that the particular individual is suffering from a Turner syndrome. Okay. And now, can you guess the total number of chromosomes over here? Because one chromosome is deleted or one chromosome is dropped off. So we say that the total chromosome count here will be 44. That will be the autosomes plus one sex chromosome, that is 45 chromosomes. The total number of chromosomes will be 45. Now, instead in that particular male, if this male, instead of an extra Y, if that particular male individual is getting an extra X chromosome, okay, so the genotype will be two X chromosome and one Y chromosome. So XXY. Now in this XXY genome, genotype, it is known as the Klinefelter syndrome. And this particular male will be showing feminine characters. The individual will be a man, will be a male, but it will be showing the feminine characters. So, because of the extra X chromosome. Now, the total number of chromosome count here will be 47. Okay. Now, sometimes instead of one extra X chromosome, an individual might get two extra X chromosomes. So, that situation or that condition is known as the extreme Klinefelter syndrome. That is it has got three X chromosome and one Y chromosome. So, the total number of chromosomes here will be 44 plus 4, that is 48 chromosomes. Out of these 48 chromosomes, 44 are the autosomes, 3 are the X chromosomes, and 1 is the Y chromosome. Now, this extreme Klinefelter syndrome individual will also show the same symptoms as the individual suffering from a Klinefelter syndrome, but more than that. It is even more uh, extreme, even more severe. Now let's discuss about the autosomal aberrations. Till now we have discussed the aberrations, that is the change in the number of count in the sex chromosome. But what if there is a change in count in the autosomes? So we have, in total we have 23 pairs of chromosomes. Out of these 23 pairs, 22 pairs are our autosomes and one pair is the sex chromosome. So out of these 22 pairs, so if in the 21st, so in the 21st chromosome, instead of a pair, we have got three chromosomes. So the 21 chromosome number 21, normally it will be having two copies, but sometimes there is trisomy that a third copy is also formed. So we say that it is trisomy. When there is three copies of a particular chromosome instead of two copies, we call it trisomy. Then we say that trisomy of 21st chromosome gives us the Down syndrome. Okay. The recent movie Sitare Zameen Par, as it is trying to create awareness regarding this Down syndrome in the society. Okay. Then there is Edwards syndrome. This Edwards syndrome is caused by trisomy of the 18th chromosome. So that is the three copies of the 18th chromosome. Instead of two copies, we have got three copies of 18th chromosome. And then there is Patau syndrome, which is caused by trisomy of the 13th chromosome. So that is the 13th chromosome has three copies instead of normal two copies. Okay. So that was our discussion regarding the aberrations, the sex chromosome aberrations as well as the autosomal aberrations. Now we move to a small topic, that is the mitochondrial replacement therapy or the three parent baby technology. Okay. Now we know that during the process of fertilization, okay, in fertilization there is fusion of male gamete and the female gamete. Okay, there is fusion between the sperm and the egg. But in actually during fertilization, it is only the sperm nucleus that enters the egg cell and fuses with it. Okay. So during the fertilization, so if this is the egg cell or the ovum and this is the nucleus of the egg cell. Now, so if this sperm is coming, so only the nucleus of this particular sperm cells, because sperm is also a cell, so that means it is also be having the chromosome nucleus in it, and that nucleus will be housing 23 chromosomes, because these the egg cell as well as the sperm cell both are ploid cells. So there will be 23 chromosomes inside the female egg nucleus, and there will be 23 chromosomes inside the male sperm nucleus. Okay, so all the DNA will be there, but so this only this part is fusing with this particular nucleus and it is giving us the zygote. A zygote with a nucleus having 46 chromosomes. That is 23 coming from the father and 23 chromosome coming from the mother. Okay. Now, if only the nucleus is entering and fusing with the nucleus of the egg cell, then in the zygote we can say that the zygote is getting the all the cytoplasm in this zygote cell is essentially the zygote of the egg cell. Yes. So therefore the total cytoplasm of zygote is from egg cell. Okay, because only the nucleus is traveling to the nucleus of the only the nucleus is entering the egg cell to fuse and to fertilize. Therefore, the total cytoplasm of the zygote thus formed, it has the cytoplasm of the egg cell. Now, in the egg cell, there is mitochondria. So, this is mitochondria. So, now this particular zygote, it will also have the mitochondria of the egg cell itself. Okay. So, the mitochondria of the father is not getting involved or is not passing down into the mitochondria of the zygote or the child. So, the zygote, if this zygote, the mitochondria it has, the mitochondria's, uh, the zygote's mitochondria has been passed down by the egg cell only. Okay, it is the mitochondria of the egg cell that is now behaving now as the mitochondria of the zygote. So, and we know that mitochondria also contain DNA. Okay, even in a very minuscule amount, but as compared to the DNA that is present in the nucleus, but the mitochondria do possess certain so it do possess some amount of DNA in it. Okay, DNA is the molecule. Now, the mutations, the changes in this mitochondrial DNA. Okay, the DNA that is present in mitochondria, we call it mitochondrial DNA. Now, certain mutations, certain changes, random changes in this mitochondrial DNA can give rise to diseases such as the Parkinson's, Alzheimer's, etc. Okay, so now we want to prevent the this mitochondrial DNA, the mutated mitochondrial DNA. we want to prevent this particular from mitochondrial DNA to pass down to the offspring. So how can we do that? So now it can be done by using what we call the mitochondrial replacement therapy. So there is mitochondrial replacement therapy. So in the zygote, so if this is a zygote, we have 23, 23. So 23 chromosomes of egg. Then we have 23 chromosomes of sperm nucleus. And along with this, in the zygote, we have small amount of this mitochondrial DNA as well. Now, the term mitochondrial replacement therapy suggests now instead of the mitochondria, instead of getting the mitochondria from the egg cell, now we remove this particular mutated mitochondrial DNA. Okay, and now we are inserting the mitochondrial DNA from a donor. So mitochondrial DNA is inserted from a donor egg. Okay, so we have in this particular zygote 23 chromosomes that are coming from the egg nucleus, 23 chromosomes that are coming from the sperm nucleus. But the mitochondrial DNA, we have removed the mutated mitochondrial DNA and has now inserted the mitochondrial DNA that we have got from a donor egg. Okay, so if some female is donating her eggs, then from that particular donated egg cell, we are taking out the mitochondrial DNA and then putting it there. So we can say that this particular, the child that will, the zygote will form a baby. Okay, the zygote will ultimately form a baby, this zygote. So we can call it this baby as a three-parent baby because three genetic parents are involved. Okay, the genes or the genetics or the genome, the entire genome is coming up from three different individuals or three different parents. That is one from father, one from mother, and the mitochondrial DNA is coming from the donor. That is why it is called, it is also known as the three parent baby technology. And till now, United Kingdom is the first and the only country in the world. United Kingdom is the first and only country in the world that has approved of this mitochondrial replacement therapy or the three parent baby technology. Okay. Now let's discuss the applications of the application part of the biotechnology. So this mitochondrial DNA, mitochondrial replacement therapy is also an application of biotechnology only. The stem cell technology is also a part of the applications of biotechnology. Now we discuss about the human genome project. Now this particular human genome project, it was a scientific project that was devised in 1990s to sequence the entire or complete human genome. Now, what is genome? The question might be that what is a genome? So, genome is the haploid genetic content of a cell. the haploid genetic content of a cell, that means one copy. Haploid means half. That is, we are diploid. So we are taking only, we have two copies. We are diploid. But in the human genome definition, we take the only the haploid genetic content. So one copy of total genetic material of a cell. So that is the human genome. Okay. One copy of the total genetic material of a cell. Okay. Now, so this particular project was concluded in 2003. And what were the observations? The observations are startling that 95%, okay, the 95% of DNA in a human cell is the junk DNA. And junk DNA, we have discussed in last session that it is a DNA that is non-functional. That is, it is not coding for any of the proteins or any of the substances. And it is not coding for any traits in the body as well. Okay. This junk DNA is the non-coding, and we also call it the selfish DNA. Okay. And we have also learned that mutations in this junk DNA can give rise to newer genes and which can form the basis of evolution. Okay. Now, and there is another thing that there are around 30,000, approximately, we have also discussed this thing, that approximately 30,000 genes are there in the complete genome. So the complete genome, the entire length of the human DNA, okay, there is only 30,000 genes that are 30,000 particular sequences that are functional or that are coding for something useful in the body. So we have 30,000 genes in 23 chromosomes. Okay. We know that the genome is the haploid genetic content. So we have 46 chromosomes. If we are taking haploid, that means we are taking the half number of chromosomes, that is the 23 chromosomes. Now, in the entire genome, there are 30,000, approximately 30,000 genes. So we can say that in the 23 chromosomes, there are 30,000 genes. So in total, in the 46 chromosome, in the entire cell, okay, entire cell has two copies of each chromosome. So the 46 chromosomes are there. So we can say that in a cell, there will be 60,000 genes in a cell. Okay, 60,000 genes in a pair of, in 23 pairs of chromosomes. Okay. Now we discuss the applications. So we have found that, okay, we have studied the whole genome, and we have found the entire sequence of DNA, sequence of nucleotides on the DNA. Now what? How can we apply? So this application of Human Genome Project are inevitable, like they are the application of the biotechnology itself. Okay. So what are the applications that we can use in designer embryo or designer baby program? So in the embryonic stage itself, we can edit the gene, or we can edit the genome to get the desired qualities or desired characteristics in the embryo or the ultimately the baby. Then there is pharmacogenomics. There is gene cloning and whole animal cloning, gene therapy, genetic engineering, DNA fingerprinting. Okay, so these are the applications of the Human Genome Project as well as the uh, applications of biotechnology. Now let's discuss about these applications one by one. So the designer embryo or the designer baby technology, like it involves two things. Firstly, we have to see whether that a particular embryo is not having any harmful mutations. So what we do is we run a pre-implantation embryo scanning and screening for pre-implantation genetic diagnosis. Pre-implantation means implanting the implantation is the process by which the embryo is embedded in the uterine wall for the further development of fetus. Okay. So before implantation, what we have to see in the embryo, that we have to screen, we have to diagnose that there shall not be any mutations, any harmful mutations. So we have to look for the harmful mutations. Now, if we find that there are certain harmful mutations, then we can use the genetic editing tools, the gene editing tools such as CRISPR-Cas9 to edit, okay, to edit the genes, to edit that particular genome before the implantation itself. So, firstly, we have diagnosed the uh, harmful mutations. Then we try to rectify these harmful mutations by using the gene editing tools such as CRISPR-Cas9. So that is the designer embryo technology. Now, this designer embryo technology, uh, it is allowed in certain countries only up to the for the research purpose. Okay, and no embryo older than the 14 days. Okay, no embryo shall be used which is beyond the 14 days. So if you are doing the research on this designer embryo, so within 14 days that embryo needs to be discarded. Okay, now second is the pharmacogenomics. Okay, now what is this pharmacogenomics? As the name suggests, it is made up of two words, that is pharmacology, which is the study of drugs, and the genomics. Yes, genomics, you know, the study of entire genome, and pharmacology is the study of drugs. So this pharmacogenomics is a new area which studies the role of genome in drug response, like how our own genome is responsible for our response towards a particular drug. So suppose I have taken a drug, how my body is gonna react or respond to that particular drug will mostly be determined by the my genetic makeup or my genome. So this pharmacogenomics studies that particular role of genome in the drug response. Now, drugs can be of two types. There can be active drugs and there can be prodrugs. So the active drugs are active when we take them. So they are in an activated form. So they are in an activated form when we take them, and in our body, the drug metabolism take place. So there is breakdown of that particular drug molecule, and then that after the metabolism, that particular drug becomes inactivated. So from active when taken to inactive after the metabolism. This is the active drugs. Okay. Then there are prodrugs. So prodrugs are just the opposite of the active drugs. That is, they are inactive when we take them and they get activated on drug metabolism. So they are inactive when we take them, and after the drug metabolism, after metabolism or breaking down in the body, they become active. Okay. Now, these two types of drugs, so any of the any drug can be made into an active form and as well as in the pro form. Now, to what population or to which individual do these uh, drugs target? So, suppose if a person is having poor drug metabolism, so if the metabolism is poor, okay, so in that particular individual, we have to give the active drugs because we are giving the active form of the drug, and that active form of drug is now acting in the body. Okay, then because the metabolism is poor, had we given him or her the pro drug, the poor metabolism would not have been able to convert the inactive pro drug into the active drug. Okay, so for poor metabolizers or the person or individual who has the poor drug metabolism in their body, they take the active drugs. For people who have very rapid metabolism, the ultra rapid metabolism of drugs in their body, they are required to take this pro drugs. Okay, because the ultra rapid metabolism, if their metabolism is really very rapid and they take the active drug, okay, then They have very rapid metabolism, so quickly their body will metabolize that particular drug and the drug will become inactive. So the prolonged action of drug will not be there. So that is why we give them the inactive drug or the pro drug, and their body then rapidly in a quick manner they metabolize that particular drug, and the individual now has the active drug in his or her system. So that is the concept with that was active drug and the pro drug. Now, another application of this biotechnology is the animal cloning. Okay. Now, what is a clone? So, clone we say is a copy. Okay. In general English terms, we say that a clone is a copy. Okay. But in biological terms, in the scientific term, we say that clone is the child of a single genetic parent. Okay. We know that a normal animal is a child of two genetic parents. That is the male parent as well as the female parent. But when we are making a clone, so what we are doing, we are making a child out of a single parent. Okay. And how are we doing this? So the technique that we use, the technique that is used to make the clone of animals is the somatic cell nuclear transfer. Now, what is this somatic cell nuclear transfer? So as the name is suggesting, so we are transferring the nucleus of the somatic cell. So now we look at the steps of this particular technique. So we take the egg cell. We have a egg cell from a donor. So suppose if this is the egg cell from a donor. Second thing we do is we remove the nucleus. Remove the nucleus from this donated egg cell. So now we have this, we have removed this particular nucleus removed. Now we take a somatic cell. So if this is the somatic cell, if this is my somatic cell, and this is the nucleus of the somatic cell. Now we transfer this particular somatic cell into the body of the egg cell. Okay. So there is transfer of somatic cell nucleus. So this somatic cell is any cell of the body that is not a sex cell. So this somatic cell nucleus is transferred into the egg cell, and now cell, this particular egg cell with the nucleus of the somatic cell, it will behave like a zygote. Now, what do we have in a zygote? In zygote, we have a nucleus. So zygote is a cell having a nucleus. Okay. And in that particular nucleus, there are 46 chromosomes. Yes, 46 chromosomes, 23 are coming from father, 23 are coming from mother. But now we also know that the somatic cells, they are diploid cells. So that means diploid means they are having 46 chromosomes inside the nucleus, and we are transferring this entire nucleus having 46 chromosomes, we are transferring it into this particular egg cell. So now this will effectively become a zygote having 46 chromosomes in it. Okay. Now, all the other machinery will be of the egg cell only. So we can say that only a single parent. Now, when we are in the normal reproduction, the two parents are involved. So 23 are coming from one, 23 are coming from the other. 23 chromosomes coming from either of the parents. Now, there are 46 chromosomes coming from only the one parent, whoever is giving the somatic cell. Okay. So that is the single genetic parent. So this particular zygote now will develop. Okay. This cloned zygote, this is called the cloned zygote, and it will be placed in a womb of a female, and then we get a baby or the individual who is the actually the clone, the exact replica of the person whose somatic nucleus was taken. Okay. So the genetic buildup, the genetic makeup of that particular clone will be identical to the parent from which the particular somatic cell, the somatic nucleus has been taken from. Okay. The female or the mother who is carrying the child in her uterus, genetically she will won't be having any effect on that particular baby. Also, the female donor who has given her egg, also she will won't be also having any effect. She won't be having any effect on the genetic buildup of that particular clone. Okay. So clone is the child of a single genetic parent. And this technology or this technique, the name is very important. It is the somatic cell nuclear transfer. The nucleus of a somatic cell is being transferred to the egg cell. Okay. Now the other application is the gene cloning. Till now we have discussed about the cloning, that is the making a copy of a whole individual. Now how do we make a copy of a gene? And why do we want to make a copy of gene? Because with the copy of gene, we have to express certain things so that we can make or we can produce certain biological goods. Okay. So now pay attention, this gene cloning. So suppose this is a healthy human cell. So this is a healthy human cell, and this is the for just for diagram purposes, I have drawn it this way. So and this is the DNA. Okay. And this is the DNA, and in DNA we have this is the part of DNA, that is it is a gene that is coding for insulin. So we call it the insulin gene. Okay. Now with the help of the gene editing technology, gene editing techniques, we cut, we take out this particular gene and we place that particular gene, or we transfer that particular gene, cut and transfer that particular gene into the bacterial DNA. So this is a bacterial cell. This is a bacterial cell. This is the bacterial DNA. And now we have transferred this particular gene, that is the insulin gene, into this uh, bacterial DNA. Now what will happen? This particular bacterial cell now will start producing, now will it will code for insulin production. So we allow this bacteria. So ultimately what we'll allow is like this particular bacterial cell with a copy of gene in it. We allow it to multiply. Okay. So in a bacterial cell, we have introduced this human insulin gene, and we now let that particular bacteria to multiply. Now, if that particular bacteria is now multiplying, so that means we are getting more and more copies of this insulin gene. Okay. And ultimately, what will we be getting is the more and more insulin. Okay. So that particular, so we say that the gene, we are making the copies of that particular gene, and ultimately the why we are making the copies of that gene, because we need large quantities of insulin or the desired product. Okay. Now there are multiple examples or the multiple what I can say the applications of this gene cloning. Okay. So applications or better will be the examples of gene cloning. One example with that we have seen is the artificial production of insulin. And we can also artificially produce, what we can artificially produce, we can produce the clotting factors. Clotting factors are the substances that are required for the proper clotting of blood. Now, if these clotting factors are absent, then we have a disease that is known as the hemophilia. So, we can artificially produce the clotting factors to treat hemophilia. And artificial production of insulin is to treat the diabetes. Okay, so hemophilia is of two types. We have hemophilia A and we have hemophilia B. So hemophilia A happens when there is eighth clotting factor is absent. So absence of eighth clotting factor gives us the hemophilia A. Now, if there is absence of a clotting factor, so now we can artificially produce the clotting factor using the gene cloning method and can treat this particular hemophilia A. The hemophilia B is due to the absence of the ninth clotting factor. So, ninth clotting factor is absent in hemophilia B, and just as hemophilia A, we can artificially produce this ninth clotting factor using gene cloning and can treat this particular hemophilia B. Then we can produce the growth hormone. We can produce the growth hormone, and like what will this growth hormone do? It will be used to treat the dwarfism in certain individuals. Okay, so individuals remain dwarf when they have a lack or they have insufficient growth hormone in their body. So by artificially making this growth hormone using the gene cloning, we can treat the dwarfism. Okay, and also we can use this particular gene cloning technique, gene cloning method to make the recombinant vaccines. Okay. Now, the next application of this biotechnology is the production or the making of genetically modified crops. That is GM crops, genetically modified crops. And the prominent examples of GM crops is the BT cotton, BT brinjal, and GM mustard. So these are the three genetically modified crops. So now, what is the methodology? Like, how are we making these genetically modified crops? We take the example of BT cotton, and then we'll also discuss the GM mustard as well. So this BT cotton, this BT stands for Bacillus thuringiensis. The Bacillus thuringiensis, this is a soil bacteria. That is, it is a bacteria found in soil. Bacillus thuringiensis. Now, this bacteria, Bacillus thuringiensis, it has a BT gene in it. Okay, and this BT gene makes a BT toxin. So, if this is the bacteria, Bacillus thuringiensis, in this bacteria we have a BT gene. Now, this BT gene is coding for or is expressing a BT toxin. Now, what this BT toxin does it, and this BT toxin is poisonous to bollworms. And please note that the bollworms, especially the pink bollworms, the army bollworms, these are the pests for the cotton plant. Okay, for the cotton crop, these are the pests. So the larvae of these bollworms, they feed on the cotton ball in which we have cotton fibers. So ultimately, it is destroying the the cotton crop. So we have to eliminate these larvae, or we have to eliminate these bollworms. So we know that there is this BT toxin. If we can introduce that in the cotton plant. If we introduce this Bt toxin into the cotton plant, then the larva will die and the crop will be safe. So now how can we make sure that the cotton plant is making this Bt toxin? For that, we have to introduce the Bt gene into the seed or into the genome of the cotton plant. So, by the genetic editing method, the gene editing method, we cut and paste by using gene editing tools. We cut and paste this particular gene. Okay, so for cutting, we use the enzymes called nucleases. So, in gene editing, when we are cutting certain piece of nucleotide or the DNA. So, for cutting, we use the enzymes called the nucleases. So these act as the molecular scissors. So we cut down the desired genes, cut down the desired DNA using this nucleases enzymes. And there are certain other enzymes that are known as ligases, which the function of ligases is to that they paste. Okay, uh, uh, they adhere or they paste two different uh, uh, sequence of DNA. Okay, so two pieces of DNA are joined together by the ligases and a piece of DNA is cut through by the nucleases. So they act as the scissor and glue in gene editing. So scissor is the nucleases and glue is the ligase. So nuclease, we cut the DNA. In ligase, we bind or we join the two DNA strands. Okay. Now, we take this Bt gene, we cut the Bt gene from the bacterial DNA and paste it using the ligase enzyme into the cotton seed cells. Now these cotton seed cells will give the Bt cotton plant, and this Bt cotton plant will be of high yield. It will give high yield and it will be pest resistant. Okay, we call it HYPR variety. And now the second is the genetically modified mustard. So we know that in the earlier classes we have learned that the mustard plant, okay, that we call sarso in the northern belt, this mustard plant is self-pollinating. That is, it has both the male as well as the female reproductive part in the same flower. It is self-pollinating. And when a plant is self-pollinating, so it becomes really difficult to make the hybrids. Okay. Hybrid is crossing the two different varieties of a plant. If we cross the two different varieties of a plant, we get the hybrid, and this process is called the hybridization. So, if the plant is self-pollinating, then the fertilization takes place within that particular flower, and it becomes really difficult for us to make hybrids out of this. So, the first task here is to that we have to stop this particular self-pollination, okay, and has to ensure that we are cross-pollinating it, because cross-pollination enables us to do the hybridization or to make hybrids. So to do this, we insert three genes. So three genes are added to this mustard variety, three genes to make the flower or to make the plant cross-pollinating, so that we can get the uh, hybrids, desired hybrids. So what this Barnase gene does, it confers the male sterility, thus preventing self-pollination. So in the same flower, we have both the male parts and the female parts, but it is making the male part sterile. So that particular flower will now effectively behave as a female flower. Now we can if it is behaving as a female flower, now we can cross it with any of the desired uh, male variety. So this is the function of the Barnase. Now, this bar gene, it confers the herbicide tolerance. Okay, it makes the plant uh, tolerant to the herbicide, that is sprayed into the fields. Third is the bar star gene. Now, we have uh, made the male part sterile and we have got the hybrid. We have got the hybrid, that is the hybrid plant, that is our product. But now we want that particular hybrid, that desired hybrid, we want that particular desired hybrid to now self-pollinate. Okay, because it is a desired hybrid. Now we want to make more and more copies of itself. Okay, now we want that particular hybrid to multiply. So now we have to restore that particular male fertility that we have taken from using the Barnase gene. So this bar star gene, it is now added, that restores the male fertility. So this combination of Barnase and bar star. So Barnase is conferring the male, or it is taking away the male sterility, male fertility from the parent flower. But when we are getting the hybrid flower, in the hybrid we want it to be self-pollinating, then we are restoring it the male fertility using the bar star gene. Okay. Now, these three genes, the source of the Bt toxin, we know the Bt gene was the Bacillus thuringiensis. Now, these three genes are also sourced from a bacteria, that is Bacillus amyloliquefaciens. Okay, this Bacillus amyloliquefaciens is the bacteria from which we are getting these three genes and embedding it into the uh, genome of the self-pollinating mustard plant and getting the desired hybrid. Now, the final application is the transgenic animals. So, this is the last application that we'll be discussing today, the transgenic animals. Now, transgenic animals are animals that are modified genetically, or the genome of that particular animal is modified artificially. So, we can take the example of a cow. We can take the example of sheep. Okay. So, from animals, we get certain important uh, products having economic value. So, cow gives us milk, sheep gives us wool. Okay. Now, in milk, there is the prominent protein that is known as the casein protein. So, if the casein protein is higher, then the milk production from the cow will be higher. And the wool is made up of a protein called keratin. Okay. Now, if this is a animal DNA, it can be a cow DNA or it can be a sheep DNA. And we have this desired gene. So that is, it can be the casein gene, okay, and it can also be the keratin gene. Okay. So for cow, it can be the casein gene, or for sheep, it can be the keratin gene. So this is the desired gene. And what it is doing, this desired gene is expressing for our desired products. And desired products are the milk and the wool. That is, the desired products here will be casein and keratin, which ultimately will give us milk and wool. So, okay, we know that, but we have to maximize, we have to enhance the production. So, how are we going to enhance the production over there? We introduce certain extra gene. We introduce a sequence of DNA, okay, and we call it the enhancer, and it is also called the regulatory gene. We add this enhancer to the genome. Now, what it will do, it will boost, it will enhance the production of that particular desired product, and the product will ultimately give us the economic value. Okay. So we are modifying the genome of we are modifying the genetics of the particular animal by adding an enhancer in order to enhance the yield that we are getting from that particular animal. So that is the concept of transgenic animals. Okay. So that's all for today's class. We have discussed the applications of biotechnology. There are a couple of applications left that we can discuss in the subsequent classes.